Good afternoon, friends. On behalf of the Nutrition Society of India Mumbai Chapter, it's my pleasure to welcome all of you to today's webinar. Friends, there are two reasons for us to conduct today's webinar. Number one, as an association of qualified nutritionists and dietetic experts, we wish to contribute scientific information towards creating awareness on certain aspects of diabetes mellitus in the community. And secondly, this is one of the topics requested by majority of the participants of our previous webinars. So today we are here with you. The increasing global burden of nutrition-related non-communicable diseases calls for sustainable preventive approaches that would encourage dietary change at an individual as well as population level. The Human Genome Project has indicated a scope for hypothetical potential for Nutrigenomics that could provide information on the interaction between nutrients and other dietary bioactives with the genome at the molecular level to understand how specific nutrients or dietary regimes may affect human health, thus paving way for personalized nutrition. To enlighten us today on the interrelationship between diet and genes in terms of nutrigenetics and nutrigenomics, we have with us an eminent expert, Dr. Vimal Karani. I would like to share few very impressive professional details of Dr. Karani with all of you. Dr. Karani is an associate professor in Nutrigenetics and Nutrigenomics in the University of Reading, UK. He has joined the University of Reading after his postdoctoral training at the Medical Research Council Epidemiology Unit and University College London. He has received advanced training in epidemiology at the London School of Hygiene and Tropical Medicine. He has published extensively in the areas of nutrigenetics and non-communicable diseases, and his 75 publications in peer-reviewed reviewed journals and also several book chapters are highly appreciated worldwide. He has received significant global media attention for his work, not only of publications, but also of his deliberations on this topic, on nutrition, lifestyle, and cardiometabolic diseases. He has procured excellent research grant for various research projects that he has collaborated, both in the capacity of principal investigator as well as co-investigator. You can see the list of sponsoring agencies for his research, which proves the high quality of research Dr. Vimal has been conducting. And I must mention to you that he is a part of large-scale multi-centric collaborative project called GENUNE, that is on gene-nutrient interactions. His primary interests are also the interests of Nutrition Society of India, that is investigation of gene-nutrient interactions on metabolic and cardiovascular disease-related outcomes. He has a long-term goal, that is to use the findings from observational studies to carry out human intervention studies towards developing personalized nutrition strategies through industrial collaboration. Excellent quality of a researcher to bring together his research work, his lab research work, industry, and the community. And today, he will be speaking on feeding your DNA, the science behind nutrigenetics and nutrigenomics. I proudly welcome Dr. Vimal Karani. Dr. Karani, over to you.
Thank you so much for a wonderful introduction. So let me share my slides. Are you able to see my slides?
Yes, yes, doctor. You can go.
Okay. A very good morning, a very good afternoon, and good evening to everyone who have joined this session from various parts of the world. So today I'm going to talk to you all about the science behind Nutrigenetics and Nutrigenomics. And first of all, thanks to the organizing committee of the Nutrition Society of India Mumbai Chapter for inviting me to give this lecture on Nutrigenetics and Nutrigenomics. So I'm going to give you some very basic aspects about Nutrigenetics and Nutrigenomics and the science behind how the nutrigenetic findings could be used for implementing personalized nutrition. So to begin with, what is nutrigenetics and what is nutrigenomics? So nutrigenetics refers to the study of the impact of the DNA sequence variations on chronic disease outcomes in response to a particular diet. For example, if I'm consuming a high carbohydrate diet and I've got a high genetic risk, so what is the risk of developing a disease such as obesity? So I'm trying to understand whether my genetic risk and my dietary intake is interacting and contributing to the development of the disease. And that area of research is what you call as nutrigenetics or gene-diet interaction. You're understanding the impact, a combined effect, an interactive effect between the genetic and the dietary factors in relation to the development of a particular disease such as obesity or diabetes or a cardiovascular disease. Whereas nutrigenomics, as a name implicates, omics means expression, genomics is gene expression, and nutrigenomics is where you're looking at the impact of the nutrient on gene expression. So that is nutrigenomics. So findings from nutrigenetics and nutrigenomics are essential to implement personalized nutrition. So in nutrigenomics, you're trying to understand the impact of how a particular nutrient can influence the expression of the DNA to RNA and RNA to proteins. And the findings from nutrigenetics and nutrigenomics are essential to implement personalized nutrition where an optimum diet could be tailored according to an individual's genetic makeup. So now let's have a look at these gene-diet interactions and gene-nutrient interactions and how these interactions can contribute to the development of a particular disease. So in terms of leading these gene-diet interactions, what we need is a lot of population-based studies. So I'm leading two large collaborative studies. One is called as the DCARDI Collaboration. DCARDI stands for Vitamin D, Cardiovascular and Diabetes-related traits, which the focus of which is mainly on vitamin D, and which is being funded by the British Heart Foundation. And to date, we have nearly 35 studies comprising more than 100,000 individuals, and from different parts of the western parts of the world, looking at the US, UK, and the European countries. And we also have access to the UK Biobank, which has got 500,000 people. And with access to all the consortia-based studies, we have a sample size of nearly 1.9 million individuals. And these are some of the publications from the DCARDI Collaboration. So the DCARDI Collaboration is focusing mainly on the developed countries. So what is happening? What is the status of nutrigenetics in developing countries or lower middle income countries? So I'm leading another large-scale collaborative network, which is called as the Genuine Collaboration. And Genuine stands for the gene-nutrient interaction. The main objective of this collaboration is basically to implement nutrigenetic studies in contrasting ethnic groups. And these are the countries in which the Genuine Collaboration has implemented nutrigenetic studies. And what you see in the brackets of all these countries are basically the funding organizations, the funding bodies, which have enabled us to implement such nutrigenetic studies in all these countries. And these are some of the publications from the field of nutrigenetics from the Genuine Collaboration. So I'm going to briefly touch upon the findings from the DCARDI Collaboration and the Genuine Collaboration. So starting with the DCARDI Collaboration, as mentioned before, DCARDI stands for Vitamin D and Cardiometabolic Disease Traits. So the main question of the DCARDI Collaboration is whether vitamin D status is associated with cardiometabolic diseases such as obesity or diabetes or cardiovascular disease or any other chronic disease outcomes. So in terms of examining the association between an exposure and outcome, it's always quite difficult because of high level of confounding and the reverse causation. So we used a a novel genetic epidemiological tool, which is called as a Mendelian randomization. So what is Mendelian randomization? That is, we use genetic variations as instruments which will mimic the exposure. So the association, the causal link is established between the genetic variation and the outcome of interest. For example, if I want to look at the association between vitamin D and obesity, I'll be looking at the genetic variants for vitamin D and test for the association of the genetic variants with obesity. And that's what we do as part of Mendelian randomization. So starting with obesity, you know that obesity is one of the main risk factors for the development of various disease outcomes, and one of which is type 2 diabetes and cardiovascular disease and even cancer. So start taking obesity as a main lead. We kicked off with this Mendelian randomization. So our first question was whether obesity was causing vitamin D deficiency or whether vitamin D deficiency was causing obesity. So in order to understand this bidirectional analysis, we implemented this Mendelian randomization approach, and we identified that obesity leads to vitamin D deficiency. That is, for every 10% increase in BMI, there was a significant decrease. There was 4.2% decrease in 25-hydroxyvitamin D concentrations in our body. That is, if your BMI increases, your vitamin D levels goes down. So this study was published in 2013. And next, we went on to test whether vitamin D deficiency was associated with cardiovascular disease. And this study was done in nearly 155,000 individuals from different parts of the UK, US, and Europe. And we identified that for every 10% increase in vitamin D concentrations, there was 8% decrease in the risk of developing hypertension. So vitamin D levels, if you improve your vitamin D status, you can overcome the genetic risk of cardiovascular disease. And that study was published in Lancet in 2015. And another study which we published a couple of years ago, where we looked at 7 different types of cancers, looking at the prostate cancer, uterine cancer, ovarian cancer, breast cancer, and various other types of cancer. And this study was done in nearly 200,000 individuals, like 100,000 people with cancers and 100,000 people who were controls. And we did a case control study design, and we identified that higher levels of 25-hydroxyvitamin D concentration can protect you from the development of prostate cancer, whereas we did not find any causal link for the other types of cancer. So this is another study which was published in 2017. And independently, there was a study that was done, which is a longitudinal study. We looked at the pregnant mothers. We looked at the vitamin D levels during pregnancy. And also, we looked at the newborn babies of these pregnant mothers. And we wanted to know whether vitamin D deficiency during pregnancy was associated with any of the fetal growth. And we identified that pregnant mothers who had vitamin D deficiency during pregnancy, they gave birth to babies which had small head circumference. And again, another a genetic risk of vitamin D deficiency was established, and which was clearly shown in the next generation. And quite recently, last year, we carried out a large-scale Mendelian randomization phenome-wide association approach, which was published in Lancet. Here, our main goal was to understand whether obesity is showing a causal association with various disease outcomes. And we looked at 925 disease outcomes. And out of the 925 disease outcomes, we identified that there were 33 diseases, including diabetes and circulatory diseases and endocrine diseases, skin diseases. There were 33 disease outcomes which showed a causal association. And a couple of years ago, we also carried out a study, which is a study linking depression and obesity. So there's always a question, is it depression that is causing obesity, or is it obesity that is causing depression? And this study clearly identified the fact that it's actually the depression that is causing obesity, and which was shown on the basis of a genetic association. And currently we are doing all other analysis looking at the milk intake, how the dairy intake or the milk intake can have an impact on obesity and cardiovascular disease outcomes, and trying to identify the links between the lipids and vitamin D deficiency and inflammatory markers and vitamin D deficiency. So in a nutshell, briefly, the DCARDI Collaboration clearly highlights the fact that it all starts with the depression. Your mental health is a key to your success. So if you keep yourself positive and stay happy, that can prevent the development of obesity. And obesity, if you can prevent obesity, then you can prevent the development of all other diseases. So the take-home message is stay happy and healthy. So now let's have a look at the nutrigenetic studies in developed and developing countries. So I'm going to give you some examples of the studies I carried out in the European population. So this study was done as part of the Diogenes Consortium. Diogenes stands for the diet, obesity, and the genes, which is basically a pan-European program that focuses on the impact of the genetic and the dietary factors in relation to weight gain. Yes, weight gain. So where we looked at 11,000 European individuals from 5 different European countries, where we pooled all the samples together and we carried out a meta-analysis, and all these individuals were followed up for 3.5 years. And we found the gene protein intake interaction on weight gain. So in this graph, you can see on the x-axis, you find the protein intake. On the y-axis, you find the weight change per year. And the 3 lines are basically people with high genetic risk, moderate genetic risk, and a very low genetic risk. And you can see that individuals consuming high amounts of protein-rich foods and those who carry a high genetic risk, they had significantly higher levels of weight gain compared to people who have a low genetic risk. So which means that if you are a European and you have a high genetic risk and you consume high amounts of protein-rich foods, then you're likely to gain more weight compared to a person who has got a low genetic risk. So the best way to prevent weight gain in those who have high genetic risk will be lowering the protein intake that is less than 17% of the total energy intake. That will be an effective way to prevent weight gain in those who have a high genetic risk. So this clearly shows that, of course, the protein intake is definitely essential, and proteins are very much essential for the normal maintenance of the physiological system, but anything in excess can have a negative impact on your health outcome. So that's the take-home message from this particular study. And the second study, again, it's part of these European individuals from the Diogenes Consortium. And here we looked at the Mediterranean diet instead of the protein. So you know that the Mediterranean diet is quite rich in omega-3 fatty acids, fruits, vegetables, and olive oil. So someone consuming a Mediterranean food is likely to overcome the genetic risk of weight gain. And that's exactly what we have identified in this large number of samples. So in this graph on the x-axis, you find the Mediterranean diet score. So which means that higher the score, higher the consumption of the Mediterranean foods. On the y-axis, you find the weight change per year. And the 3 lines that you see, basically, the low genetic risk, moderate genetic risk, and the high genetic risk. And we found that individuals who have a high genetic risk, we know that people who have high genetic risk, they are likely to gain more weight. But here you can see that people who have high genetic risk, by consuming high amounts of the Mediterranean foods, they were able to reduce their weight gain. So which means that even if you have a high genetic risk, it doesn't mean that you're destined to become diseased. You can still overcome that genetic risk by consuming a healthy diet, such as high amounts of Mediterranean foods as part of your dietary pattern. So this again comes back to the same fact that you can overcome the genetic risk by adopting a healthy lifestyle, that is consuming healthy foods. So now let's have a look at the genuine collaboration. So genuine stands for the gene-nutrient interaction. So all the examples that I've given you so far, they've all been carried out in the Caucasian population, that is mainly in the developed countries. Now let's have a look at what is happening in the lower middle-income countries as part of the genuine collaboration. The picture over here, it clearly highlights how your genetic makeup interacts with your dietary intake and contributes to the implementation of the personalized diet based on your genetic makeup. So the first nutrigenetic study in an Asian Indian population. So I'm going to give you an example of the study that we did in Asian Indians, which is the first nutrigenetic study of its kind. And that was published a few years ago. So this study was done in 1,000 people with type 2 diabetes and 1,000 people who were controls. And we did a case control study design. And we identified a very interesting interaction between the genetic risk and carbohydrate intake on the risk of development of obesity. So the red bars that you see, basically individuals with high genetic risk. On the x-axis, you find the carbohydrate intake. On the x-axis, you find the carbohydrate intake in tertiles. On the y-axis, you find the risk of developing obesity. And you can see that individuals consuming high amounts of carbohydrate-rich foods, that is consuming more than 500 grams of carbohydrate per day and carrying a high genetic risk, and if they happen to be an Asian Indian, then they have 2.46 times increased risk of developing obesity. That is, being an Indian and high genetic risk and consuming high amounts of carbohydrate, that is more than 500 grams of carbohydrate per day, that can increase the genetic risk of obesity, that is 2.46 times increased risk of obesity. And this study was done in South Indian population, and those who are living in South India, they know very well that they do consume significant amounts of carbohydrate, irrespective of whether it's a spicy food or when it comes down to dessert, there's increased consumption of the carbohydrate. The main source of energy comes from the carbohydrates. And the next first nutrigenetic study was done in the Asian Sri Lankan population. And again, the Sri Lankans, they do consume a diet which is not very different from that of the South Indian population. They do consume significant amounts of carbohydrate. So we were not surprised by this finding, where we found an interaction between the genetic factors and the carbohydrate intake on waist-hip ratio. Again, waist-hip ratio is one of the risk factors for the development of central obesity. So here on this, in this graph on the x-axis, you find the tertiles of carbohydrate, low, medium, and high. On the y-axis, you find the waist-hip ratio, an indicator of central obesity. And the red bar is a high obesity genetic risk, and the white bar with dots, which is a low obesity genetic risk. And you can see that individuals consuming high amounts of carbohydrate-rich foods and carrying a high obesity genetic risk, they had significantly higher levels of waist to hip ratio. So which means that if you are Sri Lankan and if you have a high genetic risk and you're consuming high amounts of carbohydrate-rich foods, that is consuming more than 450 grams of carbohydrate per day, then you're going to have a significantly higher levels of waist hip ratio compared to those who have a low genetic risk. So which clearly shows that consuming an unhealthy diet can increase the genetic risk of obesity. And that's what was seen for the Sri Lankan population as well. And again, when you're looking at the diet of the Sri Lankan population, it's again almost the same. It's rich in carbohydrates, but the way they eat and the way they cook is slightly different from the South Indian diet. And what you see over here, a kind of a dessert, which is called as a vachalapam, which is rich in carbs and also rich in saturated fatty acids. And the third study was the first nutrigenetic study in a Southeast Asian population, which was done in Indonesian population. There are several islands in Indonesia, and we looked at one of the islands, which is the West Sumatra Island. The reason why we went for this particular island is that in this particular island, there's a community of people. They are called Minangkabau community. And Minangkabau is the world's largest matrilineal community. And we wanted to look at this particular population and we wanted to identify the gene-diet interaction, because this particular community of people, there was very high prevalence of cardiovascular disease. So we wanted to understand what could be the driving factor, and we wanted to know the underlying gene-diet interactions, which could be one of the risk factors for the development of the cardiovascular disease. So in this population, we found an interaction with the protein intake, which was to a surprise, because Indonesians, of course, they come under the Southeast Asian population and they do consume significant amount of rice. So we were expecting an interaction with carbohydrate, but surprisingly, we found an interaction with the protein intake. So on the x-axis, you find the tertiles of protein intake, low, medium, and high consumption of protein intake. On the y-axis, you find the waist circumference, again, an indicator of central obesity. The red bar is high obesity genetic risk, and the white bar with dots is a low obesity genetic risk. And if you look at those individuals who are consuming low amounts of protein-rich foods and carrying a high genetic risk, they had significantly lower levels of waist circumference. So how do you explain this? Well, if you look at those individuals who have a high obesity genetic risk, they are likely to have higher waist circumference. But what we identified was those individuals who have high obesity genetic risk, but consuming a low protein diet, they were able to overcome the genetic risk of having higher waist circumference. So I'm not saying that low protein diet is healthy, but in this particular graph, you can see that low, medium, and high consumption of protein-rich foods. So people who are on the highest tertile, they were consuming high amounts of protein, that is nearly about 140 grams of protein per day, which is double the amount of the dietary recommendations for protein intake for the Indonesian population. So the low protein diet group is basically consuming about 70 to 75 grams of protein per day, which is almost the same as the dietary recommendation. So as long as the Indonesian population is following the dietary recommendations for the protein intake, then they can protect themselves from overcoming the genetic risk of central obesity. And we have to understand the fact the kind of diet they eat. The Minangkabau population, they have a very unique and a traditional diet, which is called as the rendang. So they eat the beef rendang and the goat rendang, which is like very common food that's being consumed almost every other day. And the consumption of the animal protein is very, very high in this particular community of people. And the next example is about the first nutrigenetic study in the Western Asian population, which is the Turkish population. Of course, the Turkish diet is unique, but it is almost the same as the Southeast Asian population because the consumption of the animal protein is again very high for the Turkish population. And we found an interaction with the protein intake again. So in this graph on the x-axis, you find the tertiles of protein intake. On the y-axis, you find the central obesity. And the red bars are those individuals with high genetic risk. And you can see that Turkish people who have high genetic risk and consuming high amounts of protein-rich food, that is consuming more than 135 grams of protein per day, they had 3.3 times increased risk of central obesity. But of course, the kind of diet they consume is almost the same, but the way they eat and the way they cook the meat is completely different from the way the Southeast Asian population is consuming the animal protein. So the take-home message from the Genuine Collaboration is that the Genuine Collaboration provides the first evidence of all these gene-diet interactions in all these Asian populations. So in terms of understanding the interactions between the genes and diet in relation to obesity, when you look at all these populations, whether West Asian or South Asian or Southeast Asian, there are different dietary patterns which are consistently seen. If you take the South Asian, India and Sri Lanka, it's basically the carbohydrate diet, which is the driving force for increasing the genetic risk of obesity. Whereas if you look at the West Asian population or the Southeast Asian population, it is a high protein diet, which is actually driving the association between the genes and obesity. So in terms of implementing personalized nutrition services to treat obesity or diabetes or cardiovascular disease, we definitely need evidence from all these studies because the findings from one population cannot be used for the findings from the other population. If you want to implement personalized nutrition for the Indian population, then you will have to use the findings from the Indian population. You cannot use the findings from the UK population and implement that in the Indian population. So that is one of the most important things that we are trying to convey and bring awareness as part of the Genuine Collaboration. And if you're interested in knowing more about the Genuine Collaboration, you can go into this publication, which was published earlier this year. So now let's move on to Nutrigenomics. We have seen a lot of gene-diet interactions, which is Nutrigenetics. Now let's move on to Nutrigenomics. So Nutrigenomics is nothing but the study of the role of nutrients in gene expression, how a particular nutrient can influence your gene expression level. So for example, if you're consuming any particular diet and the food that you consume has got different nutrients, it could be micronutrients, it could be macronutrients, any kind of a nutrient. So the nutrient needs to be absorbed by all the cells in your body, right? So the nutrient goes inside the cell through the cell membrane and it reaches the cytoplasm. And there is a molecule that is waiting for the nutrient to enter into the cytoplasm. And that molecule is nothing but the transcription factor. So there's a specific transcription factor for a specific nutrient. So the main function, there are two important functions of the transcription factor. One is to bind to the nutrient. It has to recognize its nutrient and bind to the nutrient. And the second important function is to take the nutrient to the nucleus where the DNA is. So the transcription factor, along with the nutrient, it goes and binds to the region called promoter in the gene. It binds to the promoter and regulates the gene expression. When I say regulating the gene expression, it means that the DNA is converted to RNA, and RNA is converted to proteins, and proteins are further metabolized to produce the metabolites in the body. So in response to a particular nutrient, there are certain RNA molecules which are produced, and there are certain proteins which are expressed, and there are certain metabolites that are produced. So the study of the impact of the nutrient on the mRNA expression is called transcriptomics. And the study of the impact of the nutrient on the protein expression is proteomics. And the study of the impact of the nutrient on metabolite expression is metabolomics. So these areas of research, genomics, transcriptomics, proteomics, and metabolomics, they all come under one big umbrella, which is the nutrigenomics. So now we have a very important question. Why do we need all these omics platforms? Why do we need to understand the gene expression? Well, if you're consuming a high carbohydrate diet, for example, there are certain genes which are overexpressed or underexpressed. Or if I'm consuming a low protein diet, very, very low protein diet, there are certain genes which are underexpressed or downregulated or upregulated. So there are some changes in the gene expression due in response to the kind of diet you're consuming. So if you're on an unhealthy diet, that is going to have an impact on the gene expression profile. So if you understand that you have a defect in your gene expression profile, then if you can rectify your dietary pattern at an earlier stage, that will give you an ability to prevent the development of the disease at a later stage. So prevention is better than treating or managing the disease. So that's the whole point of understanding nutrigenomics. So these differences in the gene expression markers, they serve as a molecular biomarkers. Once you know that these are the molecular biomarkers, you can use these differences in the gene expression profile. You can rectify it by modifying your lifestyle, and you can prevent the development of the disease. How do you prevent the development of the disease? By implementing the personalized meal plan, which is suitable for an individual according to that individual's genetic makeup and the gene expression profile. So it all starts with the kind of the food and the diet that you're consuming. All the foods and the diet that you're consuming has got all different kinds of nutrients, whether it's macronutrients or micronutrients or minerals or fatty acids or vitamins or antioxidants. What do? They go inside your body. They need to be absorbed by the cells, and they influence the gene expression in different tissues, whether it could be adipocytes or it could be liver cells, or it could be pancreatic cells, or it could be brain cells. So eventually it is affecting the expression of the genes in different tissues, and that is what is required for maintaining your glucose homeostasis. So a healthy diet will maintain the glucose homeostasis, whereas an unhealthy diet can disrupt the glucose homeostasis, and that can result in the development of diseases such as type 2 diabetes. For example, we say the foods which are high in phosphorus can lead to increased blood pressure and pulse rate. So how do we know that these foods are having an impact on blood pressure and pulse rate? So there are various mechanisms which are involved. So some people might prefer foods which are high in salt, and some people would prefer gluten-free diets, and some people will prefer foods that are high in sugar, and some people would prefer to eat something without sugar. So there are different food choices and food preferences which are entirely controlled by your genes. But when it comes down to kind of restriction on a particular kind of a diet, then which is what we call as a calorie restricted diets, or sometimes people who undergo fasting, there are different mechanisms happening inside your body. That is, you're disrupting the normal physiological system. So let's have a look at what is the science behind the fasting mechanism, that is intermittent fasting or periodic fasting or religious fasting. So there's always kind of a positive view towards thinking about the intermittent fasting. So people who fast, they have a positive impact. So that's kind of a belief. Of course, there are a lot of evidence, scientific evidence saying that fasting or the caloric restricted diets do have a significant impact on the positive health outcomes. So let's have a look at some of the scientific evidence. So one study clearly showed that the caloric restricted diet can decrease the IGF-1 signaling pathway. IGF-1 is the insulin growth factor 1 pathway, and that can reduce the production of the IGF-1 levels. And we know that there is an evidence that links reduced production of the IGF-1 level and the anti-aging mechanism. So which means that studies have shown that the caloric restricted diets can make you look younger than before. And another study has shown that caloric restriction can decrease the production of the inflammatory markers, and which means that there's an impact on the anti-inflammatory effects. And another impact is about the caloric restricted diets on adipogenesis, how the adipocytes divide. So that happens when you restrict the number of calories you consume, that can activate a co-activator, a molecule called PGC1 alpha. So that functions right in the beginning of the pathway. So this activates this co-activator, and that can regulate the process of adipogenesis. So if you look at this particular the figure, it shows that the PGC1 alpha, it works right in the upstream of the pathway, which will go and bind to the DNA and regulate the gene expression, which is required for the adipogenesis. So this caloric restricted diets can have a positive impact on activating the PGC1 alpha. And another important study is where they showed that the caloric restriction can lower the levels of oxidative damage to the proteins or lipids or DNA in response to stress. So basically, all these studies basically shows that the caloric restricted diets do have a positive impact on health outcomes. But having said this, you have to understand that all these studies have been done mainly in animals. So that is exactly the reason why the science behind fasting is very, very uncertain. We really do not know what is happening in humans because the animal studies do not necessarily apply to humans. And secondly, most of the human clinical trials have been very, very short, and the longest trial so far has been only 2 years. And thirdly, most of the studies have been done in individuals aged less than 60 years, so we cannot apply all these mechanisms to people above 60 years. And then the number of organic and chemical processes, they keep the human body functioning, indicating that the human body is very complex and the humans are different from each other, not only in terms of the age or the height or weight or gender or environment and lifestyle, but also the genetic makeup. My DNA is different from that of your DNA. And lastly, the reliable evidence is harder to obtain in human studies. Even in animals, it's difficult, but compared to humans, it's much more easier in animals, but the animal studies do not necessarily apply to the human studies. And that's exactly the reason why the science behind fasting is still uncertain. And that is one of the reasons why fasting works for some people and it does not work for other people. So in terms of understanding the nutrigenetics of fasting, we still do not have a convincing evidence that the fasting will have a positive impact on overcoming the genetic risk. Definitely not, because there are studies which have shown that people who underwent the caloric restricted diets for a period of time, they actually gain more weight compared to people who do not carry a particular genetic variation. So that clearly identifies the fact that the people are different in terms of the genetic makeup, and we need to take into consideration of the genes before applying or providing a personalized dietary service for an individual. So now, having talked about the DNA, the human genome, which has got 3 billion nucleotides and which carries 40,000 genes, and these 40,000 genes are coding for nearly 150,000 transcripts, and these transcripts are coding for several thousands and thousands of proteins, and which are further metabolized to all these metabolites. So we have nearly about 3,000 metabolites in our body, and the levels of the metabolites and the composition of the metabolites vary from one individual to another individual. So the levels of my metabolites might be different from that of yours because of the kind of diet we are consuming. And the levels of these metabolites are an indication of a risk factor for the development of various chronic diseases such as obesity or diabetes. So this study is what you call as metabolomics or metabolic profiling, which comes under the umbrella of Nutrigenomics. So briefly touching upon what is this metabolomics, what is metabolic profiling? So you can do these kind of studies either in humans or animals, depending upon the kind of sample you have. So you can use a blood sample or serum or plasma or urine or fecal or breast milk. So depending upon the type of question you have, sorry. And you can use any kind of a metabolic profiling technology. Either you can use a mass spec or NMR or any kind of chromatographic technique. But the output is really, really complicated because a lot of multivariate regression analysis that you'll have to carry out, and comparing several hundreds of people and looking at different metabolites, you do get different peaks for each metabolite, and understanding the levels of the metabolites across different people, that is the key for understanding the molecular basis of the health and disease. And you will get to know that some people will have higher levels of a particular type of a metabolite, which might be a risk factor for the development of type 2 diabetes. And you use these levels to identify the risk of developing the disease, and accordingly, you can correct the levels of the metabolites based on your lifestyle modification. So next one is about the impact of the diet on epigenetic modification. So we have seen nutrigenetics, nutrigenomics, and under which we have seen the metabolomics. So all these are more of a genetic mechanisms. That is, you're looking at the DNA, and in the DNA, you have different DNA sequence variations, the genetic variations, and which are conferring different levels of genetic risk. But epigenetics is completely different, which is not looking at the DNA changes. So epigenetics is basically the heritable changes in the gene expression, which can make a gene either active or inactive. Or in other words, epigenetics is basically the study of the change in the phenotype without the change in the genotype. You're not changing the genotype. Genotype is nothing but the genetic representation of an individual, whereas a phenotype is a physical representation of an individual. So the epigenetics is otherwise called as a non-genetic mechanism. It has got nothing to do with the DNA, but you're looking at the impact of the DNA. That is, you're looking at the change in the phenotype rather than the change in the genotype. There are two kinds of epigenetic modifications. One is the DNA methylation, where the methyl group is added onto the cytosine, and that becomes a methylated cytosine. And the other one is a histone modification, where the histone octamer, which is bound by the DNA molecule, it undergoes some modifications such as hyperacetylation and methylation. So these are the two important epigenetic modifications. And let's have a look at what is this DNA methylation. So this DNA methylation occurs mainly in the promoter region. So the promoter region is where the transcription factors bind and regulate the gene expression. So the transcription factors can go and bind to the promoter only when the promoter is not methylated. So say, for example, in this particular figure, you can see that the transcription factors are binding to the promoter because the sequence is not methylated, and unmethylated DNA will allow the binding of the transcription factor, and that will result in the gene expression. The other option is where the promoter region is methylated. For example, if you're consuming a methyl-rich diet, the methyl groups can go and bind to these promoters, right? So that will prevent the binding of the transcription factors, and that can result in gene silencing. So this is how a gene expression is regulated in your body in response to the kind of diet you're consuming. So you cannot have the gene expression to occur throughout the day. At times, the gene silencing will have to occur to maintain normal glucose homeostasis. If the gene silencing is disrupted, then that can affect the glucose homeostasis, and you can develop obesity or diabetes. So let me explain a classic example of this agouti mouse model, which is an animal study, which clearly explains the fact how the epigenetic modifications can be reversed. If you look at the DNA genetic variations, you're born with the genetic variations. Until the last day of your life, you're going to live with that genetic risk. You cannot change your genetic risk, but you can overcome your genetic risk by modifying your lifestyle. Whereas epigenetic modifications, you can modify, you can change, you can reverse the epigenetic modification, but you cannot change the genetic variation. You need to understand the difference between the epigenetics and genetics. So let me explain that with the agouti mouse model. There's a gene called as the agouti gene. So where there is a promoter region which can get methylated, or it can stay or remain unmethylated. So this agouti gene will code for a particular protein. That particular protein is the one that will determine the fur color of the mouse. So if the region is methylated, if the promoter is methylated, then that will result in gene silencing, and that will result in the healthy mouse with brown fur. If the promoter region is not methylated, that will increase the expression, that is overexpression of the agouti gene, and that will result in a mouse with yellow fur, and the mouse is likely to become obese and develops diabetes during adulthood. But this is where the twist is. So these yellow mice with yellow fur, if they undergo pregnancy, and during pregnancy, you're providing a methyl-rich diet, that can reverse the epigenetic modification. That is, the methyl groups can be added to the unmethylated region, and that can result in a generation of mice which are healthy with a brown fur. So that's why I'm saying you can reverse the epigenetic modifications, but the genetic variations cannot be changed. So this picture basically shows how the methyl groups are added on to their cytosine and resulting in the methylated cytosine. And the next epigenetic modification is a histone modification. So as I said before, the histone modification, the histone exists as an octamer. There are 8 subunits, H1, H2, H3, and different subunits are there. And there are about 8 subunits. And within these 8 subunits, there are different regions in the histones which undergo hyperacetylation and methylation, which is allowing the gene to get expressed or not expressed. So these histones are basically wound by the DNA molecule. So the first step is basically where hyperacetylation takes place in the histone 3 and histone 4. This hyperacetylation will open up the DNA structure and it allows the gene expression. So that is basically where you're allowing the expression of the genes. But if you want to bring the gene silencing, then you need the methylation of the lysine residues in this H3 protein. And this is how the methylation occurs and makes the DNA tightly coiled around the histone proteins and preventing the expression of the genes. That is, if it's tightly coiled, the transcription factors cannot bind and allow the gene expression. But if the structure is open, it allows the gene expression by binding of the transcription factor. So this is how the histone modification can regulate the gene expression. So there are a lot of studies that have shown a clear link between epigenetics between the maternal diet and fetal growth. So I'm going to give you some scientific evidence of how the maternal diet can have an impact on the fetal growth. So that's exactly the reason why we say the first 1,000 days plays a very important role in a child's life. So unbalanced maternal diet during pregnancy has been shown to have an impact on the fetal growth, and that can remain postnatally, and that can lead to the development of cardiovascular disease risk in adulthood. And the seasonal variations in maternal methyl donor nutrient intake during the periconceptional period can have an impact on the abnormalities in the maternal plasma biomarkers, and which can have a result in the changes in the methylation of certain genes in the infants postnatally. And there's also another study which has shown that the maternal obesity during pregnancy can result in the DNA methylation changes during birth, and also it can remain postnatally. And obesity and diabetes during pregnancy, that is gestational diabetes, can result in differential methylation, which was observed in the DNA from the umbilical cord blood of the offspring, and also in 4 to 5-year-olds and 9 to 16-year-olds. So all these studies clearly highlight the epigenetic link between maternal diet and the fetal growth. And recently, there was a study which showed that the micronutrient deficiency during pregnancy can also have an influence on the DNA methylation and having an impact on the fetal growth. So I'm going to give you an example of a study where we looked at the micronutrient deficiency. That is, we looked at the vitamin D deficiency during pregnancy, and let's see what happened in terms of the fetal growth. So this is called as a vitamin D pregnant mother cohort study, which was done in the Indonesian population. And we identified that pregnant mothers who had high genetic risk, and those in the 3rd trimester, they had significantly lower levels of 25-hydroxy vitamin D concentrations. So those who have high genetic risk, they have significantly lower levels of vitamin D levels. Now let's see the babies that were born to these pregnant mothers, and we identified that these pregnant mothers who had high genetic risk and lower levels of vitamin D, they gave birth to babies which had a smaller head circumference. And we all know that small head circumference has been shown to be associated with development of cardiovascular disease in adulthood. This was not an epigenetic study, but it is more of a longitudinal study, which is giving a kind of a hint for the link between the micronutrient deficiency during pregnancy, which has got an impact on the fetal growth or the newborn anthropometric measurements. Well, there is definitely a link, but that needs to be identified whether it could be an epigenetic modification. So to prevent such changes or abnormalities in the fetal growth, so the foods rich in methyl donors will be the right choice. So because what you do and what you eat in the first 1,000 days, it makes a very big difference for the rest of your life. And the last part of my talk, which is going to be about the impact of the diet on the gut microbiome. So we have seen how the food that we are consuming, it interacts with your genetic variations. and interacting with your gene expression and influencing your metabolite levels and also affecting your epigenetic modifications. Now let's have a look at the gut microbiome. The gut microbiome plays a very important role in maintaining a healthy physiological system, because the food that you're consuming, it goes inside the gut, and in your intestine, there are several microorganisms that are present, not just bacteria, but also viruses, phages, and yeast, and they do play a significant role in contributing to maintaining your immunity levels and maintaining your mental health, and also providing an anaerobic environment for the growth of the microorganisms. So let's have a look at the role of gut microbiota in health and disease. So sorry about this busy slide. So in this slide, I'll just try to make it very simple for you. So on the left-hand side, you see the red colored rectangles. On the right-hand side, you see the blue color rectangles. So looking at the left-hand side, you see that the impact of the unhealthy foods, that is, if you're consuming foods that are rich in saturated fatty acids or high in sugar or excessive protein consumption or consuming antibiotics regularly, they can all have an impact on the gut microbiome. That is, they can reduce the production of the short-chain fatty acids. There are three important short-chain fatty acids, that is the acetic acid, propionic acid, and the butyric acid. All these three short-chain fatty acids play a very important role in maintaining your immune system and also maintaining your mental health and also providing the anaerobic environment for the growth of the bacteria. And in addition, these unhealthy foods can also increase the production of the TMAO. What is that? Trimethylamine oxide. So when you're consuming a diet that is high in meat and dairy products, then there's actually a chemical called as a trimethylamine, which undergoes oxidation, and that can result in the production of the trimethylamine N oxide. And high levels of trimethylamine N oxide has been shown to be associated with increased risk of cardiovascular disease. So reduced production of the short-chain fatty acids and increased production of the trimethylamine oxide can affect your gut. That is, can induce inflammation and can increase the risk of infection and can reduce your cognitive levels and also increase the risk of developing type 2 diabetes. On the other hand, if you're consuming a diet that is high in fiber-rich foods or taking a lot of probiotics, that can have a positive impact on your gut microbiome. That is, it can increase the production of the short-chain fatty acids, increasing the production of the antioxidants, and which can basically have an indirect impact on improving your insulin sensitivity and decreasing the insulin resistance, improving your lipid metabolic activities, and reducing the gut inflammation and reducing the risk of the infection. That is exactly the reason why a lot of studies now, they are coming up, which are showing a link between the gut microbiome and COVID-19. So nourishing your gut microbiome is equally important as nourishing your DNA as well. So all these studies are definitely required for the implementation of the personalized nutrition, where an optimum diet could be developed for an individual according to their genetic makeup. Your DNA is everything. The DNA makes you all very unique in different ways. So in terms of translating the findings from DNA to personalized nutrition, so starting right from the diet, so the diet that you're consuming, the food, it goes and interacts with your DNA, which is in the nucleus, and that area of research is what we have seen as nutrigenetics. And the next area of research is where it's interacting with the metabolites, and we have seen that as Nutrimetabolomics, looking at the metabolite diet interaction. And the other one is where we have seen that the diet can interact with your gut microbiome, and that area of research is Metagenomics. And finally, we have also seen the epigenetic modification, how the diet can influence the epigenetic markers. And all these four areas of research, that is, all these functional activities, or whether you're looking at the gene diet interaction or metabolite diet interaction or gut microbiome diet interaction or epigenetic modification, they are all happening simultaneously as soon as you consume any kind of food. So if you're thinking about implementing personalized nutrition, it is very much important that you need to look at it as a four-way interaction, as metagenome, hyperbolome, epigenome diet interaction, which is the one that's going to be useful for the implementation of the personalized nutrition. So from nutrigenetics to the prevention of the non-communicable diseases, starting right from nutrigenetics, nutrigenomics, proteomics, metabolomics, and also the gut microbiome, the area of research is what you call as the metagenomics. So it has to be more about the implementation of a holistic approach in terms of preventing the development of the non-communicable diseases such as type 2 diabetes. So before I finish, one last slide. So these emerging human nutrition trends are the ones which are going to shape the future of the food industry. These human nutrition trends are nothing but the nutrigenetics, nutrigenomics, metabolomics, gut microbiome, epigenetic markers, and all these are the ones which are going to shape the future of food industry. A lot of researchers are working in various fields of nutrigenetics and nutrigenomics, and there's a lot of evidence available, but still kind of a mystery. We need to solve the puzzle. Where do we start? Where is the starting point? The starting point is understanding your genetic risk. At this point of time, it is very difficult to bring everything on board and trying to implement a personalized diet. But as a starting point, the beginning of this entire vision is basically in the hands of the gene testing centers, where they are trying to identify your genetic risk and trying to provide a personalized diet according to your DNA. So that is where I see as a starting point of this prevention of diabetes or any other cardiometabolic disease related outcome. So on this note, I would like to thank all the DCARDI collaborators, and thanks to all the genuine collaborators. And finally, thank you all so much for listening to my talk. And if you have any questions, you can drop me an email at v.karani@reading.ac.uk. And I'm available on Facebook, LinkedIn, Instagram, and Twitter as Vimal Karani. And thank you once again. And thanks to the organizing committee again for inviting me to give this lecture, and happy to take any questions.
Thank you so much, Dr. Vimal. It was really an intellectual feast listening to you. I really have no words to appreciate you. I just want to say very briefly that very strong evidence-based information shared in a very crisp manner, so meticulously by you through excellently prepared slides. So I have no more words to describe your presentation. It was indeed a great learning experience for all of us. Thank you so much for that. Since we are running short of time, I'll quickly take just few questions. As you have shared your email ID, the participants can always send you their queries. So first, I wish to begin with my question only to you.
Yes, sure.
So the information that you had shared from DCARDI Collaboration as well as other Nutrigenetics projects is excellent information. It really opens our eyes towards, like, you need to look at another angle as well. Okay, so in that, in the current scenario, the obesity in India is also being described as thin obesity. Okay, not everybody is having high BMI or high WHR or WC. Okay, so in that kind of situation, so what is the role of gene testing or nutrigenomics there? How do you explain that?
Well, that's a very good question. In fact, like, we are moving towards a situation where people who are obese can still be metabolically stable, and people who are not obese can be metabolically unstable. And there is also a project that is going on where we are trying to look at those genetic markers which play an important role, people who are still healthy, because BMI is a kind of a very crude measure. So
Yes.
we cannot go anything by the BMI, even though there are a lot of genome-wide association scans that have identified gene variants for BMI, body fat percentage, and waist circumference. And they are not consistent. There are certain genes which play only for BMI. There are certain genes which play only for waist circumference. So we need to dissect out certain genes which play an important role, which is influencing only BMI and only body fat percentage, and try to understand how they can respond to the kind of diet we are consuming.
Okay. So it's actually a very difficult area to understand, because maybe probably the association between body components and the gene interactions, maybe the association between that might throw some light on that. Is there a possibility?
Of course, like, there aren't many studies that have looked at the gene-diet interactions in relation to people who are obese and metabolically stable, because as I said, the functioning of the body is very complex, and trying to understand such a metabolic instability in terms of how that can have an influence in relation to your diet is very complicated.
Yes, Dr.
Maybe a dietary intervention study will be able to explain that.
That's great. And you had shared a lot of information on vitamin D. Actually, in fact, you began with that, and it is because vitamin D is very famous now. It's very popular nutrient currently. Everybody is paying a lot of attention to that. So here, your information on maternal vitamin D deficiency resulting in babies with low head circumference. So that's a very interesting piece of information. But we are actually coming across some piece of information on vitamin D deficiency. It may not be always related to the consumption of vitamin D through diet. It is like basically endogenously synthesized. Okay, but however, probably there could be some metabolic errors due to which the vitamin D synthesis is not normal in some individuals. Probably this could be one of the reasons for vitamin D deficiency. So here, in case if the pregnant woman is vitamin D deficient, okay, because of this kind of problem, and is there a possibility that the newborn also shares that genetic defect?
Well, it depends, because like what I'm talking about is mostly genetic variations which can exist as a low or moderate or high genetic risk. And you know that the mother contributes like one copy of the gene and the father contributes the other copy of the gene. So the child basically gets 50% chance of getting the genetic defect from the mother. So there's definitely 50% chance of inheriting that metabolic defect. But then again, it could be complemented by the remaining 50% that is coming from the father, which could be a healthy version of the gene as well. But having said that, the vitamin D deficiency, that vitamin D contribution from the diet is very, very less.
Very poor, yes.
Yeah, so it's mainly the main source of energy is basically from the sunlight exposure. That is the main thing. So if you look at the Indonesian women, they have like sunlight throughout the year, even though they have rainy season. The sunlight is not an issue at all, but which looks like it could be a defect in the synthesis pathway or the metabolism of the vitamin D. So that is something that needs to be explored further.
Yes, that needs to be explored for, because like even in India, we don't have problem with sunlight. Of course, exposure is becoming an issue, but otherwise there is no dearth of sunlight here, but still there is increasing prevalence of D deficiency. So let's move on ahead with one kind of very strong revelation from your studies, that is about high protein intake. Because now currently in the area of nutritional management or dietary management, high protein is actually revolving around very strongly, because majority of the people are protein deficient, or quality wise the protein is not good quality. So there are so many concerns, ifs and buts revolving around protein consumption. But your study is showing high protein intake, like some negative aspects of that, like association with WHR or vagera vagera. So that is what is being shown. So here, of course, I know you have disclaimed it, like saying that you don't recommend low protein consumption that way. That is understood. But is it that source specific? Is it depending on sources of protein? Because the examples which you have shared are either from saturated fat containing meat products, or red meat, or preparation of cooking, cooking method of cooking. Are these the variables that are contributing more towards this probably? Is there any possibility of that?
Well, actually, if you want to consume a high proteinaceous diet, then the most important thing is you should have that ability to burn those calories as well. So people think that if I consume a high protein diet, which is healthy for me, but high protein diet is, technically speaking, it's not healthy. You need to stick on to the dietary recommendations, what is required for your body, depending upon your ethnicity. And each population, each ethnic group, or each country has got their own dietary recommendations. Yes. So especially with regards to the Indonesian population, the dietary recommendation was only 70 to 75 grams of protein per day. And they were consuming the low protein diet was actually the normal protein diet. Whereas the high protein was on the extreme high consumption of the animal protein. So they are not consuming any kind of like plant protein as far as I know. So it's mainly the animal protein which they are consuming. So that is something that needs to be reduced. And I don't see Indonesians to be highly physically active individuals, except for maybe 10 or 20% of the population who might be very active, or people who are involved in the field of sports. So definitely, if you want to consume high proteinaceous diets, then you need to exercise that much. So it's more into the muscle mass and the muscle building when it comes down to protein. It's not like carbs or fats.
Exactly, because that high protein could be converted to fat again.
Yeah, exactly.
And you have rightly said mental health is the key of holistic health. So which we, even in one of the webinars that we have conducted on World Mental Health Day, Even the experts reiterated it, and it's a very well-known fact now. So now there is one question related to that. What epigenetic change does depression cause, which results in obesity? Because you have said that it is basically depression that is leading to obesity, making it a cause for other non-communicable diseases or other conditions. So this question is very interesting actually. How do you declare that depression is causing some epigenetic change resulting in obesity?
See, the question is it the depression that is causing epigenetic change, or is it the epigenetic modifications which are causing depression? So eventually, everything is determined by the kind of diet you're consuming.
Okay.
So the kind of study that we did was like looking at the relationship between whether depression was leading to obesity or obesity leading to depression. There we used genetic markers as instruments. We did not look at the epigenetic markers. It was more like a cross-sectional study rather than a longitudinal study to understand the causal link. So we did not look at the epigenetic modifications there, because epigenetic modifications are more of a non-genetic mechanism,
yes,
rather than a genetic aspects. Yes.
That you have explained very well. It was indeed like eye-opener for many people who do not understand the difference between epigenetics and nutrigenomics kind of.
Exactly,
it was
yes.
very well explained. So it was mostly from observational studies.
Yes.
It was mostly
observational, but at the same time, a large sample. As you had seen, we had about like nearly 1.9 million people and across different parts of the world. Yes.
And the last question I would like to take for you is, what are the ethical considerations in gene testing?
Well, the most important, yeah, of course, definitely. Like a lot of people are trying to practice nutrigenetic testing and trying to implement personalized nutrition without having getting a proper qualification in the field of nutrigenetics and nutrigenomics, which is completely unethical. There are very few centers in India. I mean, of course, I'm not sure whether Janani has joined. So she's leading a genome wellness center where she's practicing nutrigenetics and implementing personalized nutrition, which is very unique and very specialized in the field of nutrigenetics, and she's highly qualified to implement such services. But there are a lot of dietitians and nutritionists who have come across, and they attend a 1-day or 2-day course, and they say they're experts in nutrigenetics and nutrigenomics, and they want to implement personalized nutrition. That is definitely not the right way. It's completely unethical. And that's exactly the reason why the personalized dietary services can also lead to a dangerous outcome. So you have to be very careful in implementing such personalized dietary services. Like a person who's taking a personalized diet can be benefited from that kind of a diet, but you cannot share that diet with the other person because the other person's genetic makeup is quite unique and different from that of yours. So you have to be very careful when taking into consideration of all these aspects.
That's great. Actually, it's very important to follow ethical considerations when one is doing gene testing or using gene testing reports. Yes. Yeah, it's and in let me just check with Neha if Janani has joined. Otherwise, I'll take the opportunity of just one.
Janani has joined, ma'am.
She has joined. Okay, then we will mail you the remaining questions.
Okay.
And though I'm very enthusiastic in asking 2 more questions to you. But still, anyways, we will always mail you the questions.
Yes,
thank you so much once again on behalf of Nutrition Society of India Mumbai Chapter. We are highly grateful to you for accepting our invite readily and making it convenient for you to join us from Reading and then give this lecture. It was wonderful, Doctor.
And all the best for the remaining sessions, and have a nice day. Yeah, thank you, everyone.
Thank you so much. Now I hand over the session to Dr. Neha Sanvalka, who will be introducing Ms. Janani, expert for this next session. Dr. Neha, over to you.
Thank you so much, ma'am. Thank you, Dr. Vimal, for such an interesting and insightful session on what the basic and what actually nutrigenomics and nutrigenetics is, and what is the basic meaning of the 2 words, because a lot of us nutritionists are really unaware of what's the difference between nutrigenomics and nutrigenetics, and a lot of us tend to interchangeably use those 2 terms. So it's been a really great eye-opener for us. Welcome, Janani, to Nutrition Society of India Mumbai Chapter. I'll just quickly share your slide and introduce you to our participants. Our next speaker for the evening is Ms. Janani Tamilvanan. She is the founder and director of Institute of Nutrigenetics, and she's also the founder and Nutrigenetic specialist of Genome Wellness. For her qualification, she has done her bachelor's in food science and nutrition and food quality control from Avinash Lingam University, India. She has a double master's degree with a master's degree in foods and nutrition from Coimbatore and a master's degree in food science technology and nutrition sponsored by European Commission. She's currently pursuing her PhD. She's won numerous awards, some of them which include Be a Change Award for a emerging startup for the year of 2017, the Erasmus Mundus Scholarship Grant. She was a university gold medalist, and she's also been a vice chairman of Avinash Lingam University 2003-2004. Before she found her own company, she has served as a senior nutrigenicist for Export Life Sciences. She's been a junior nutrition consultant for FAO Rome and Columbia. She has been a research scholar for Purdue University at Department of Food Science and Food and Nutrition. She's also been a senior dietitian for VLCC Middle East Dubai and a nutrition expert and corporate trainer for Nestle India. So with her immense experience and her entire education, her forte lies majorly in nutrient interaction and latest developments in the field of nutrigenetics and nutrigenomics. And she aims at translating the scientific evidence-based knowledge into precise, personalized lifestyle recommendations for health promotion and diet-related complex disease prevention. Janani has delivered several guest lectures, and she has counseled many patients based on personalized diet with help of their genetic makeup. So today, Janani would be talking about personalized nutrition in diabetes using gene testing. Janani, we welcome you to our chapter, and I hand over the session to you for the talk.
Thank you, Neha. And it's such a wonderful evening to spend with you all in NSI, and it's good because I think organization like yours, so once they start discussing or debating about these topics, which are quite trending now. Apart from trending, I would say this is going to be the future, and it's very crucial that all of us in this field and who are yet to come, so need to be at least knowledgeable in order to take it up. So when it is there. And I'm really glad that I'm a part of this session, and thank you for the organizers, NSI Mumbai Chapter, for taking this up, taking this topic. So this is more than a passion to me, and it's quite, it's actually a kind of fascinating science to learn about what's happening within you. And I'm going to share a little bit of it today. So 30 to 45 minutes, I don't think it's enough to share even a bit of it. But I have made efforts to put it in a very concise manner. And I think it would be on the other side of the plate where Dr. Vimal covered about the research aspects, and he's very proficient in delivering research lectures, and he's more of a researcher. So when it comes to this session, I would like to make it clear it's more of a clinical practice. And we'll be more of translating the essence of science and how do we transform into the meal plates. So I think research has to be done. And on the other side, to make it implemented is what the other challenge is all about. So I'll cover the later part of it, that is more of translational as well as transforming the meal plates of clients. And especially to do with diabetes. So I'm not a diabetic specialist because I focus more on body weight regulation and obesity. But I have made some, kind of information, to concise the information related to diabetes. But you could always correlate diabetes and obesity, as well as inflammation would be the basis of it. So I'll try to give a kind of overview of the entire concept, how to use personalized nutrition in diabetes, and what is the link, what is the connection with gene testing here. So basically, the personalized nutrition approach tries to answer why do individuals respond to the same diet in a different manner. And I guess this is the question which has been in the minds of health professionals for so many decades. So we'll be confused in spite of high calorie restrictions and giving them intense exercise. So why are they not responding? Whether it's going to be body weight regulation or whether it's going to be blood glucose regulation. So we have this question. It's been there for a long time. I think the genetic testing empowers an individual with such an information where you can find answers to this question. Basically, this common question could be answered using genetic testing. And second one is genetic variability among humans contribute to the human metabolic individuality. So all of us speak that everyone is unique and everyone has certain traits that pertaining to them. But what is that unique which contributes to the human metabolic individuality was actually mentioned in the first session, where we talk about the SNPs, or the difference in the arrangement of the DNA sequence is what we refer to as genetic variability here. And as a nutritionist, it becomes very critical for you to understand this human metabolic individuality concept, because that defines how much you're different from each other and what kind of response you have to a particular food and what could be your disease predisposition. So all these are confined by this genetic variability. And this could be actually determined by a commercial gene testing. So the current scenario is we always go with a population-based guideline. So this is a very primary information that I'm giving here. But what we assume and what's the reality is these pyramids or the population-based dietary guidelines doesn't consider the variation or the concept of human metabolic individualities. And here we are transitioning from the population-based dietary guidelines to more of individualized guidelines, but we are not stopping with just the gene sequence or the DNA sequence, which we were doing few years, about 5 to 6 years. But what we are moving ahead is we are trying to consider gut microbiome, as well as he gave insights into metabolomics. So we are no longer looking into just SNPs for determining your response to certain foods. So it's going to be more of the gut microbiome metabolomics. So it's going to be more of a system biology approach where you consider all the omics technology. So initially when we talk about personalized nutrition, so we were considering only about the SNPs, but now it's going to be more about the gut microbiome. It's more about the metabolic profiling, what are the metabolites that are produced, and later it could be about the epigenome and transcriptome as well. So personalized nutrition could mean different things for different people, but it's absolutely necessary to define in what context you're meaning to say personalized nutrition. So the Human Genome Project was the basis for why we are talking about personalized nutrition today. And one of the implications of Human Genome Project is it was able to give us a blueprint of life. And as well as another implication is we were able to find out what are the genetic variations that are associated with certain diseases, and especially it includes diabetes as well. So if you're able to find out that there are certain genes that are associated with certain diseases, it's just like you go to a mechanic shop and then you repair a car and you know where is the fault, and this is what Human Genome Project has implicated. So you know which gene is causing which defect and you could actually repair it as well, because the recent technology allows you to do that also. But at a healthcare level for a dietitian or a nutritionist, so what it brings is, so you're able to predict about the predisposition of an individual, the susceptibility of an individual to a particular disease. In this case, let it be diabetes. So if I know that a particular person is predisposed or susceptible to diabetes, so probably that gives a scope of predictive as well as preventive nutrition. So this is actually a tool for any health professional who wants to use it to empower their patients or clients with information, as well as to utilize it in order to delay the onset of disease or to prevent the disease, as well as to manage it. So this is a good piece of information that we have got from the Human Genome Project, with which we are able to use it in order to have a healthy life. So this is again a question for debate. I think like on diabetes, so many people would have given you information about statistics, but the one aspect of it, what I want to ask you here, it's a question for debate is, and it also emphasizes the need for personalized approach. So far, all these statistics in the world by different institutional authorities, it always says the numbers double or triple with years. So there's no statistics that talks about decline in obesity rate or decline in diabetes rate so far. And one of the possible reasons that we could anticipate or we could actually think of it is it could be because of ineffective strategies. So we are looking, or there must be something missing, because like we are trying to come up with medicines. We are actually coming up with different strategies to prevent and to manage diabetes or whatever disease it might be. But what is missing is what we are talking about, the genetics. So far we have been more focused on the phenotypical aspects of it. So we say that the blood monitoring has to be done, diet and lifestyle changes, and about like social factors, about stress, about mental health also it was mentioned. But what we missed is we didn't have information about the genetics or any other aspects of genomics so far. And because of the technological advancements, so we are having those information now. So it is up to us, how do we integrate the genotype and phenotype information and come up with a more comprehensive solutions. And it is also a thought process that I would like to initiate, like that I want people to think about is, so we have been more focusing on management and treatment of diseases, but it's time to do a paradigm shift where we focus more about preventive nutrition, and that's where the strength of personalized nutrition lies. So why not we start preventing a disease, the occurrence of disease, rather than after acquiring the disease, why do we need to focus more on the management and treatment? And this personalized nutrition gives you the opportunity to prevent any disease, because that is also predictive in nature, and we'll learn how do we use that information. So if you are able to predict something, how do we use that? So as you learned about the difference between nutrigenomics and nutrigenetics, so when you look at diabetes, you will have two aspects of it. So first one is we'll be looking at what are the nutrients of food, active compounds present in foods that are going to affect gene expression that is responsible for diabetes. And another aspect is we are going to talk about the genetic variations, that is SNPs, that are responsible for influencing your response to nutrients. So we need to look at both the aspects. So whether it's obesity or whether it is diabetes, so this applies. So you're not just focusing on, so I'm not just going to stop with taking a genetic test and knowing by genetic variation and then saying that this is how I respond to food. It's equally important to understand how food is influencing how my genes are expressed and how that is related to diabetes. So I'll start with nutrigenomics. So it's not possible to give you a whole view of, there are a lot of genes that are, related to diabetes. I've just mentioned few here, and I'll also try to make you understand about the connection between gene expression and diabetes. So first, let's take nutrigenomics and diabetes. So there are certain genetic markers that are responsible, and this is especially to do with epigenome. So hopefully you would have learned a few things about epigenome. So these genes, these are like the epigenetic modifications which are normally found in diabetic individuals. So just like your biomedical markers, so if there is a diabetic individual, so we'll go on with the leptin levels, insulin levels, as well as adiponectin levels. So these are few biomarkers that we test, HbA1c and so on. Likewise, there could also be epigenetic markers in the future. So let's say in diabetic individuals, so there is an increased DNA methylation that's associated with impaired insulin secretion, whereas the decreased DNA methylation, so it is with reference to a particular gene. You could find that like these are either highly expressed or there is a more of methylation, increased methylation that's associated with impaired insulin function. So this could be a biomarker, an epigenetic marker in the future, which could actually identify diabetic individuals. And the next one is it's with regards to the histone acetylation. So it's nothing but the epigenome. And there are certain ways in which the epigenome is modified, and these are certain parameters by which you could identify that these are the parameters that could be found in diabetic individuals. And plus, you're also able to connect it with the food that you eat. So if you're eating a food that's low on protein or deficient in calcium or magnesium, chromium or vitamin D, so you could find these implications there. So what do? So we can test for these gene expression of the mentioned ones. So usually it's not just 4 genes that they check for, it's more than that. So what we are talking here is about gene expression. So we are not checking for genetic variants, but this is more of gene expression profiling is done to check whether how much is expressed or whether it's expressed in that particular individual. So what do? There are certain nutrients which can act upon certain genes, and you could also give the food sources. So this is with reference to nutrigenomics. So you're identifying how certain nutrients are affecting the gene expression and what are the foods that you could give in order to combat the effects that are caused by these particular nutrients. So the next one, when we move on to the nutrigenetic aspect, so it's not something new, that is providing certain dietary recommendations based on the genetic components. But the most common condition that everyone knows is about the PKU. So this is a single gene mutation which results in a condition where there is elevated levels of phenylalanine in the blood. So once you restrict it, so you can overcome the symptoms. But diabetes is not caused by single mutation, and it is multifactorial in nature. So it is not as easy as just restricting phenylalanine food in the case of PKU. It's something beyond that, and we have different types of diabetes, and it's also quite multifactorial. So we need to really understand that. There are certain good examples that we can talk about, but what we are discussing here is about a multifactorial conditions. And in order to give precise nutrition recommendation needs a lot of in-depth understanding of the pathophysiology and the mechanisms which are the pathways that are related to diabetes mellitus. So again, when it talks about when we discuss about gene-diet interaction in diabetes, as I told you in the previous slide, so it's a multifactorial disease. So there are a lot of factors which could actually contribute to the occurrence or the onset of diabetes. When we look at this health pendulum, so you could understand that. So on the left-hand side, you have a combination of genes as well as lifestyle factors. So if this is going to be the combination, that is, you have susceptible genes which make you predisposed to diabetes along with a bad lifestyle factor, so then that is going to push your pendulum towards disease. Whereas on the other side, if you have good genetic factors along with good lifestyle factors, it's going to push the pendulum towards health. But majority of us have a combination, and that puts you somewhere in the middle, right? So this explains that this could be better explained. I often give this example. So a person having, who's a chain smoker lives longer, whereas someone who doesn't smoke, so he dies at an younger age. So it same applies with diabetes as well. So in this case, we can always say that the genetics is only one piece of the puzzle, and there are a lot more like gut microbiome, metabolomics, and transcriptomics. But the thing is, we are in the initial stage of integrating all the information, but to understand, so we need to really get into the fact is multifactorial. So there are a lot of genetic factors involved in each of the, the factors that I've mentioned here. So let's say this is a snapshot of genetic testing report, and the results are displayed here for your information. So there are certain genes tested, let's say ADPOQ gene, and you got the genotype, and they say you have a normal risk or you have a higher risk. So the red ones are highlighted, which means they carry a risk allele. And this one is the genotype, so that will be different for different individuals. So if you and me are giving the test, so this is going to be same for all of us, but this will be different for each one of us, and that only determines your risk for diabetes. So when you look here, so it's out of the 8 genetic markers tested, it's only 2 of them are risk alleles. And this is how they predict the risk. So he said, like, predicting the genetic risk could be a useful tool for understanding and treating or preventing a disease. So this is how you look at it. So you come to know whether a person is at high risk or low risk, and you can start suggesting them very precise recommendations. So how do they arrive at a particular risk score is, and I'll just tell you in brief so that you will be able to understand, and you will start validating the information. So it's not that all the genetic test reports are false or it to be validated. So some have given strong criteria, or there are a lot of reports which comes up with strong evidences, research evidences. So for you to assess it, I'm just giving few examples. So first criteria is it has to, the markers that are selected for testing, it has to be present in the population. It has to be present at least in the 5% of the population in order to be tested. So here you could see that there are a few genes which contributes to higher energy intake and lower energy expenditure. So if you could see that the risk allele frequency, it's always more than 5% in the population. And then probably you could start identifying. So you shouldn't be looking at testing for rare mutation and then like predicting a risk for obesity or diabetes. And also like people who are testing for 1 or 2 genetic markers and coming, I mean, is also not a right kind of information, because there are a lot of genes, 600 to 700 plus genetic markers associated with diabetes. So it is not right to just check for 1 gene and then to say the risk. So similarly, the risk allele is only the determinant of whether they are at risk for a particular disease or not. So if you're checking for about 4 genes, I'm just giving you an example, and if all of them are contributing to the risk, so it totals up, and then finally you say whether you're at lower risk or higher risk. So out of the 4 genes tested, it's just like your test mark. So out of the 4 questions, if you answer all the 4 right, so you get the highest mark. So similarly, you get the highest risks here. So it is the number of risk allele, and small effect is contributed by each risk allele. So it means that the more risk alleles you carry, the higher the risk for your weight gain or diabetes or blood sugar. So now you understood that there is a risk scoring method that's followed for predicting diabetes risk. At the same time, there are a lot of research evidences, especially randomized controlled trials, which have actually collated all the precision nutrition interventions for prevention as well as management of type 2 diabetes. So if you look here, so most of the research that has been done on diabetes say there is no significant intervention interaction happening, unlike obesity. So in obesity, there are at least few research evidences quite positive, saying that they were able to establish a significant interaction or a relationship, whereas in most of this, so this is the most recently published Lancet, where they did a review of all the precision nutrition intervention studies. So you could see that even the sample size are fairly good. So here you could see like 3,548. So this is about interaction between polymorphism of transcription factor 7L2 and lifestyle intervention. So what did they find out? And it was almost like 3-year study. effect of risk conferring TT genotypes at this particular SNP was stronger in the placebo group than in the lifestyle, but no significant genotype interventions. So especially to do with diabetes and in the context of gene-diet interaction, as far as my knowledge is concerned, I did a fairly good search. So it is not well studied, or it is not yet available for clinical practice to take this and then put it into practice. So we need to really have more validation done before we take these and apply it. So I can't just take one study and then say this is present in the study and I'm going to use it in my clinical day-to-day practice. So just for you to look at all these studies. So even the Pound's Loss trial is here. And they say that significant interaction between the genetic risk score is present, but that again, we can't take just this and then apply it, because it's more to do with the dietary fat on a 6 months change. So it's again the sample size we need to really look in, that gives a strength of evidence. So I would say that we need a lot more in terms of diabetes and gene diet interactions, where we could use the information as a recommendation. So similarly, what we are doing in Nutrigenetics is, so you take a marker and based on the SNPs, so here I would like to actually, show the difference. So you have you're grouping individuals into low, medium, and high tertile of dietary fiber group, and you're checking it against waist circumference. So we always, highlight about high fiber intake, especially to do with obesity or diabetes. But we need to really start looking at it. So it's again based on genotypic differences. So if you look at AA, so they actually benefit a lot than AT or TT individuals. So it's not necessary for all the diabetic individuals to have a high fiber diet. So this is a very basic interpretation. But what do we do is we need to really understand the pathways, not just the diabetic pathway, but it could be the other pathways which comes along with the report, the genetic testing report. So it is about understanding the other pathways which would be really helpful in understanding. So if you look at body weight, it's not just one linear process that happens, but it could be classified. Food energy intake. So you could see a whole lot of genes involved, starting from the hypothalamus and the genes and peptides which are related to it. And similarly, you want to actually look into the adipogenesis pathway, where you have the PPARG and the SERPP adipoq gene. I've just mentioned few. There are almost like 900 plus markers that are associated with obesity and diabetes. So when we look at the energy balance, to start off with, I've just taken this as an example. So in order to understand high energy intake, because when we look at diabetes management, so we always talk about low carb and fat, it's more about macros. And we also emphasize on micronutrients, but the main aspect is given under the macros. So let's understand how do we, how do we tweak the, the macros based on genetic testing. So you have certain genes that are contributing or influencing your higher energy intake, and there are a few genes that also determines your low energy expenditure. So you could split the energy balance or the energy homeostasis at 3 levels. So one is the cellular level, and other one is the peripheral level, and other one is the central nervous system. So the sensor, or it's kind of a cascade of reactions. So once your AMP to ADP ratio, ADP to ATP ratio falls down, so then there is a cascade of reaction which starts at the cellular level, and then it slowly progresses into the peripheral system through signaling, and once it reaches the peripheral system, it then reaches the central nervous system. So there's a kind of process that sets in. And when you look at the peripheral system, so there are certain signals that is like between the meals and tonic signals between weeks and months. So why I'm emphasizing on this pathway is, once you start understanding the pathway, we know what is the role and function of gene at the molecular level. See, this AMPK is actually the cellular energy sensor. So once the ratio drops down, that will stimulate the cascade, and you have the hunger pan. So this starts at the very basic cellular level, and once that hunger hits the signaling system, so at the peripheral levels, either you have short-term signals or the long-term ones, and that is triggered by various processes at different steps. And you have hormones, peptides, and enzymes involved in it. And basically, these hormones and peptides are nothing but proteins which are encoded again by the genes. So if you want to really learn about nutrigenetic and practice it, first it's very important to understand the molecular pathways, and it's nothing but molecular nutrition. So you have different signaling systems, and this we have learned in human nutrition. So you have hunger signals, that is episodic signals refer to those ghrelin, and the satiety signals CCK, GLP-1, and I've just mentioned a few. So these are all involved in regulating appetite and satiety. So now there is always like a kind of a homeostasis which sets in. And there is also a hedonic system. So you could find that these peptides are regulating the appetite and satiety. So what do? We understand what are the genes that are involved at each level. So if you look at the, it's a kind of homeostasis. You have the ghrelin, hunger hormone, and leptin, which makes you feel fuller. And there is also a gene which encodes these hormones. And as well as there is insulin and glucagon. And there is a gene that regulates insulin function and insulin signaling pathway, as well as glucagon, which is actually again encoded by a gene. So in every step, if you look at, there is a gene involved, and there could be a gene that could be tested, and we could figure out what kind of variant that you carry, and based on which you could customize. So these are the gene specific nutrient recommendations that we could come up with. So if you say that ADPOQ and FTO, so you could start giving adiponectin-induced foods. And we have like all research evidences for these food sources. And likewise, serotonin-inducing foods. And likewise, you have the POMC, AGRP-reducing foods. So it's all about like, how do you look at it? So understanding the pathophysiological mechanism becomes very crucial. So just to wrap this up, genes regulating appetite and satiety. So there are few genes that are regulated that just we have seen before. And how do we come up with nutrient specific recommendation is, for example, FGF21. So it actually is required for optimal insulin and glucagon. So when you look at this, betaine is found to be actually important for FGF21 gene in order to have a kind of, effective functioning of this gene. So we just give foods rich in betaine. So if you find out that an individual is having a variation in this particular gene, then it is probably that person gets benefited with betaine rich foods. And likewise, if you have a genetic variance in the retinoid receptor gene, and probably supplementing them. So because the eye is one such organ where you need to focus more on the vision when it comes to diabetes, and especially like increase in omega-3 fatty acids. And we also mention which gene we are actually targeting and which is the variant that they carry, and you start formulating gene specific recommendations. So this is the melanocortin system, which again regulates it, dopamine and GAD2, which glutathione gene. And vitamin B6 is again a cofactor which helps in the production of kappa. So what you're doing is you're not just telling them eat this food and then this is good, but you're also connecting it with the gene so that it makes it quite evident why you're giving that particular food or diet. So now to wrap it up, so we've been doing this practice protocol, following this protocol in dietary assessment. And what we are going to do is we are also going to include genes in all the, the parameters that we have been doing now. So if it's going to be diabetes, so these are some of the genes that is going to determine your BMI, influences your BMI. So when you have the results of these genes in your report, so you're going to correlate it with the BMI, WC, or waist-strip ratio. And if you are checking for HbA1c, you're not going to just talk with the quantitative measurement, but you're also going to, connect the parameter and correlate it with these genes, which are responsible or which will influence your HbA1c level. Likewise, I've just taken a snapshot, or it's not the whole entire assessment that we do, but just to give you an idea how to correlate information, I've given it here. So finally, when you look at what would be the conceptual framework of management of diabetes in the future is, so you have diet, physical activity, and medications. There's no doubt about it. And there's also like questionnaires now, digital health questionnaires, to extract information about these parameters. And what we have to do is to integrate the omics into the existing protocol, and also coupled with wearable devices, which makes it easier for you to measure as well as to record these parameters. And then we need a big data analytics, which has to be set in. So that's the way forward for precision nutrition approach, where we take into consideration all the aspects and put it precisely for someone to take it and call it as personalized nutrition. So this again, so it's one step way ahead, where we call it as precision nutrition. So thanks for the opportunity. I hope I kept my time. So I'm open for questions now.
Thank you so much, Janani, for the short and crisp talk on how diabetes, the role of nutrigenetics in diabetes. We do understand, the time is very short to cover everything into it, but still in the short time you've told us that it's not just one particular disease or one particular gene that we need to look at. We need to look at it holistically and look at all the pathways that are involved in the mechanism. So we do have a few questions for you. One of the questions is, how can Nutrigenomics help in the field to minimize metabolic disorders? As in, they want to ask, can we be applied to a general public nutrition rather than just personalized one-on-one nutrition?
I think that defeats the whole concept of it, right? So we are saying that population will not fit in, because each person is going to have individual genetic makeup and thereby individual requirement. So even when we talk about BMR or BMI, so we have set these standards based on equation that is again done based on a population. So it will take time for us to arrive at, because the BMR is also influenced by a certain group of genes. So you can't come up with saying this is going to be the formula used and this is the BMR. So it contradicts. But what I can tell you at this point of time is, if a lot of people are taking this test, so it'll become more commonly available for people to do it. So now, just because of the price factor, so imagine 10 years before, the blood test was as expensive as the genetic test. So nobody wanted to go and take a blood sugar, right? So now it's just a prick test, which is 10 rupees. And that is made possible because everyone started to do it, and everyone started to do it more frequently. But with genetic test, it's just one time, because it's never going to change.
Right. Thank you so much. So the next question is that what are the certain challenges that you face when you are planning a personalized diet for a patient based on the nutrigenetic report? So what are the most common challenges that you face when you plan these diets?
Okay, so first one is contradiction. So the genetic test could say one thing, but the actual scenario, so for example, let's take vitamin D. So the genetic test could say he will be a vitamin D sufficient, but in actual scenario, a person could have vitamin D deficiency. So how do we interpret these contradictions is one challenge, and how do we make them to understand? And second thing is, as I told you, we are not come up to a level where we can answer all the questions. So we are also slowly evolving and learning. So this aspect has to be conveyed to the the person who is willing to do the test, and we should also be transparent about being in a country like India, where we don't have research that's done in Indian population. Right. So most of the genetic testing companies take the references or take the the study results from other population. So we need to be really transparent, and sometimes it might go wrong. People actually feel offended that. So as she mentioned about ethical issues. Yeah. So we need to be really careful when it comes to ethical issues. So it's a very confidential and sensitive information. Right. So you will be in an extra risk for maintaining that confidentiality, as well as to make sure that there is accuracy in the testing, because there are all possible chances that it might go wrong, and there could be errors also when it comes to testing. So these are all certain practical difficulties, especially during lockdown. So all this was shattered because there was no no transport, and labs were closed. So you become responsible when you are practicing as a dietitian, or like you have a team. So you will be facing these. So and also with scientific evidence. So not many of them will analyze the scientific evidences, but it is the responsibility of a health professional to convey this. So not all the reports will have explicit information, right? So it's up to you to convey this to the customer or to your patient, saying that this is how you need to interpret it. So that takes a lot of time. So instead of just giving a template kind of a diet sheets in most of the hospitals, like in some places, so where they have a ready-made diet plan. So here it takes a lot of time. And also like even after putting a lot of time, it's like up to the individual, whether he will be able to sit and listen to you. So that's also.
So you mean to say that it's very important that you dedicate that time and explain every part of the nutrigenetic reporting that has been reported, and explain to the patient what each part of the report stands for. Like, that's what you're saying, right?
Yes, but again, like most of the reports are lengthy ones, at least like minimum of 50 pages. So it's not possible to, get them word by word. But once you become proficient, you know where to focus and then where to just, go ahead with it.
Which genes to prioritize based on the condition of the patient, probably. That would help you explain the things first to the patient and then maybe take it to the next step in the next sessions of counseling. Would that work?
Yes, sometimes. So to check out for the progress. So if they have signed up with you, and if you're not able to see that progress, you might sit for a while and then to understand, to analyze. Otherwise, what we do is what we also suggest others to do is to come up with a summary, just like your doctor's summary, discharge summary you see, right? So once you start summarizing it, it becomes very handy for explaining this genetics. So instead of, taking them through all the 50 to 100 pages, so you just sit with them and then like take 2 sheets and then this is what. So that'll be helpful.
So there's another interesting question, that is there a specific criteria for you to opt or for you to suggest patient to go for a personalized diet with help of a nutrigenetic report or a genetic testing, or do you follow a certain criteria and when we have to recommend to the patient that he should opt for personalized diet based on genetic testing?
So there is no set criteria, because that again is based on individual preferences. So sometimes I have told them that it's not necessary, but they'll be very willing, and then like they'll be asking us, okay, let me try it. I want to try it, and they come here with that intention to take it. So in that case, you can't go against them. But there are also cases where you would be suggesting because you're trying different things and it doesn't work out. But again, if they're not willing, so we just need to leave it. So it's up to that individual, but there is no criteria as such. See, this is an additional information for you as well as for the patient. So it's not like a mandatory thing, only with this you need to go ahead. Only thing is that if you are giving personalized nutrition, probably it's just more of empowering them, as well as is an additional bit of information for you to plan out the diet.
Right. So there's another question which says that they want to ask is, how do you integrate blood reports with gene testing? So as I think you already shared in the ABCD table, that based on the disease condition that we are looking at, probably we need to look at the gene testing, the gene that is to be correlated. Anything more that you would like to add apart from what you've already shared? Because it's
that's what I mentioned. So when it comes to vitamin D, so usually what happens is if there is a vitamin D deficiency, so you'll be listing down all the vitamin D rich foods and then giving it to them, or you would suggest them vitamin D supplements to be prescribed by a physician. So this is at the most like what we do. But when it comes to genetics and biochemical correlation, so there are like almost 2 possible outcomes. So one is it will coincide, or the other option is it contradicts. So if it coincides, or if your genetic tendency also says that you will have vitamin D deficiency, and currently that blood test is also confirming that, then probably they need to be extra cautious. So we'll be giving that. But what happens is there are certain genes that will be responsible for vitamin D deficiency. So we will be figuring out what are the genes, and we'll be coming coming up with gene-specific recommendations.
Gene-specific recommendations.
But if you don't have a genetic tendency, but it's more of something to do with the external factors. Let's say you've been to London for a few months, so where you don't get exposed, and that could be the reason. Probably you come back here, that'll get settled automatically.
So environmental factors is what you are trying to say.
Environmental factors. So it's more of a correlation. I think like once you start looking at reports, people will get hang of it. It's not a rocket science at all.
Changes in your reading. So there's one question which says that I think you've already answered this, that are there changes in your reports after some time in the results? I think you mentioned that the gene testing reports won't change after, right? So if we do the gene testing report or we don't need to repeat the gene testing report, right? It's going to be always the same test. That
when it comes to SNPs, yes, but the gene expression profiling, it will change definitely. And the epigenome that I mentioned, especially to do with diabetes or obesity, it's going to change. It's dynamic in nature. So the kind of different food that I eat, it's going to influence. Whereas the SNP genotyping test, which we call it as nutrigenetic testing, so that should be always the same. So if it is not the same, then it means that's something to do with the testing.
Either the first lab or the second lab had some mistake in either of the two labs.
Yes, but again, when you're doing it with two labs, then it's totally different, because like labs could choose different SNPs. That's not thing. But the thing is, if you're checking with the same lab, same SNP, and if it's like different, then it's something to do with the lab. So we need to really check it out.
So there's another question. I think I'll take this as the last question because I know you. So you mentioned that we study gene expression. As gene expression is a very complex phenomena, could you explain how does one study gene expression in vivo? Are there any biomarkers that we look at when we are looking at gene expression in vivo?
Yes, definitely there are. It also depends on what you want to test. So like the conditions you want to, and also like how many months, is it like a supplementation that you want to. So see, there are certain studies where you want to study the dosage. So if I'm giving turmeric for quite some time, like 6 months, so is it affecting gene expression, especially to do with cancer? So now they are recommending turmeric to cancer patients. So suppose if I want to study like whether that particular bioactive compound is bringing a change in gene expression and how much it is bringing. So it also depends on the dosage or what is the study. So it will differ from a study to study. But again, that is becoming commercialized. So it's kind of, it's a good progress that's made, not only nutrigenetic testing. So once you have the gene expression profiling, it's even more better for nutritionists. So you will have a lot of innovation happening in the field of nutrition industry.
Ma'am, I'll hand over the session to Subhadra, ma'am.
Yeah. Thank you, Neha, and thank you, Janani. It was wonderful listening to you.
Thank you.
Very nice information, and you made it very clear that the dietitians, whether they are using gene testing or the traditional conventional approach, they have to be highly knowledgeable about the pathophysiology. That is very, very important before you prescribe any diet, instead of just going ahead with the general guidelines that are available. Okay. Anyway, even before gene testing, all diabetes dietitians, they are following personalized nutritional approach, but using the other parameters of testing, the ABCDs of nutritional assessment. But this will definitely add on another angle to it and make the intervention more effective. So that is the reason why we want people to understand, there is another one, but it's an upcoming area. So I would like to actually, since we have 5 minutes, we will take 5-10 minutes more time for questions. So because there were some questions that were posed during the previous session to Dr. Vimal, which I kept it for your session, actually thinking that it may be more suitable for you. Okay. And among them, one piece of information which Dr. Vimal has shared is the, I think, cardiac study, if I remember it well. So he has shared that he has shared associations with macronutrient consumption with markers of obesity, WHR, BMI, etc. So where, of course, he has explained that very high carbohydrate intake, that is really very high carbohydrate intake, association with like no height, BMI, etc. That is very well understood. But there are other bars also on the picture where it is indicating the genetic risk. Okay, so if the genetic risk is lower or poor according to gene testing, does that mean that no, those individuals can be allowed to consume high carbohydrate? How do you follow that?
No, ma'am. It again depends on the current pattern. So if a person is already consuming more, so you can't actually prescribe. So it's always a genotype to phenotype. So that's where the research and translation practice makes a lot of difference. And also when they really say high and low in research, the studies. So when we need to really look into it, is it like the percentage or is it in grams? And which population it has been studied? Maybe the one which he studied would be like UK population, or even in so the ethnicity has to be considered, and we need to really look into the sample size also. So that's why we have like a criteria when we take all these genetic markers. So obviously we are taking the research evidence from the studies like this, where it's going to be studies. But that again, we will be validating it, and we'll be looking at the strength of evidence. So the first thing we look at is the sample size of the study. And second one is about, so if he says that people who are at low risk can consume a high carbohydrate diet, so what is actually they are referring to as high carbohydrate? So what would be the percentage? And what is the genotype that they are giving to? And for the final point is, so we can always have all these research evidences, but when we sit for diet counseling, so we will be taking the diet recall, and we will know how much they are consuming currently, and we need to correlate it with that, and then only decide how much to give them. So unless and until, we can't take the genetic report in isolation, and then like we can't just prescribe a diet just with the genetic information.
Very rightly said it is. And of course, Dr. Vimal has shared huge population. The sample size is very good, excellent. And he was of course saying very well that that doesn't mean that they can consume high amount of carbohydrate. Of course, you need to look into quantity as well as quality aspects of that in the light of the existing practices, because the graphs were showing even the other two bars, there could be some speculations in the mind of participants. So that is the reason why I want to clarify it through you. Okay, so then my last question, last but one question, let me put it that way. There was a very interesting question, that is, there is something called set point theory in obesity that all of us know very well. And disturbing that is very crucial if you really want to sustain the weight loss or maintain optimum healthy body composition. So by following gene-based diet prescription, is it possible to disturb it?
Yes, ma'am. Actually, the set point theory itself is the basis for the physiological mechanisms that I've mentioned. So when you take thermogenesis, there are certain genes that are involved in thermogenesis, and once you know the genetic variations, and then giving those gene-specific nutrients will definitely promote thermogenesis. So you would have heard about PCOS, thyroid, and all these where they speak about slow metabolism. So it's becoming a common term. So I have slow metabolism, my levels are so less, so I'm not able to lose weight. But once we start assessing the genetic markers, it's very much possible to actually bring in changes. So you're understanding at a molecular level why they're not able to lose weight, or why it is difficult for them to lose weight. So it is definitely possible. And also I have personally seen my clients, so there are a lot of things, but that needs a lot of studies. So we need to really sit and analyze. So it won't happen overnight, but it's definitely doable.
It is a possibility.
Yes, it is. So but it won't work for all the individuals.
Yes.
So that is also there.
Yeah, there are many other confounding factors that you need to keep in mind. That is very right. There are actually two more interesting questions. Because most of these diabetics are on medical treatment as well. Okay, so type 2s will be on hypoglycemic drugs and sometimes even in combination with insulin. So how do you integrate this? Like with the gene testing, with the diet, and the management of diabetes? Because the question is there any gene allele which has an effect on diabetic drugs, especially metformin?
Yes, yes. So, especially Dr. Mohan's diabetes research in Chennai. So they have precision diabetes program where they don't bring in the nutrition, like the precision nutrition into the picture, where they focus more on the risks gene. So, like if you want to find out the genetic risk of a person, and especially they deal with a lot of MODI as well as like they have they prescribe medicines based on the precision testing, gene testing. So what do they do is usually metformin and sulfonylurea drugs could actually interchanged based on the genetic test results. So that have been more effective. So if a person is not able to manage his blood sugar levels, and once you take the gene test and once you find out that you're not responsive to metformin, and the physician can immediately change him to sulfonylurea. Of course, all the other management practices will be there, is definitely needed, but changing the medicine based on the genetic tendency will become very crucial in management of diabetes. And they've already started doing it because they have a large cohort of people, and they have started. So if someone is interested to know more about precision diabetes, so they can explore that. So it's already in practice.
It's very, very important, because now after the patients start developing resistance and then by trial or error changing the drugs, that's what is happening in most of the places. And this precision management is absolutely wonderful technique.
That's so rightly said. We could avoid the trial and error. So you could also observe like people are taking metformin for almost 15 to 20 years. And then they realize that it's not working. And probably, so we can really avoid all these healthcare costs and also the health implication that they'll be facing.
Yes, very true. And last but one question, actually this I think you will be able to answer well. What are the gene mapping techniques, okay, which are used all over the world to get the genetic makeup of a person to move towards personalized nutrition? Are there any such kind of techniques for the person all over the world, kind of common ones?
A gene mapping in the sense, are you talking about the testing, or is it the mapping that they do in the research? So when you talk about gene mapping, there are different research studies that are done, genome-wide studies. So it will be like a large cohort which is done. So whereas you will have the cross-sectional studies also, like which are done like a handful, but they will be testing about like lot of parameters. So it will be very intensive. So all these gut microbiome testing, when it started, it was done only with 15 people or 20 people, the metabolomic profiling that he mentioned. So if you could be more precise on the gene mapping, what do you mean actually? So because we have all these gene map, obesity gene map, where they have figured out what are the genes that are involved in obesity. But if you could be more precise, so what are you referring with gene mapping?
Actually, this question is not very clear. This question is not very clear, but one thing is, with the information which Dr. Vimal has presented, the gene response to nutrients is highly region specific, racial specific. That's what actually he said. So when I see using all over the world, there won't be finally any commonness in understanding.
Definitely. So there's no gold standard as such for these markers. So now this is pretty unregulated. So we don't have laws or regulations to stick on to. Even there is no influence by the FDA or any health authority as such. So in that kind of a situation, you can't expect a kind of a gold standard to set in. But it will differ from industry, company to company. It's up to the health professional to get trained and become knowledgeable on this.
Because racial differences anyways are there in most of the aspects. So you can clearly say one size doesn't fit all across the world. But across the world, you leave it aside. There was one question in the previous session, that is, can we have some kind of commonness in the guidelines based on gene testing across the regions within India? Is that a possibility? Or have you come across the gene testing or proneness, from the gene testing that reports that you have seen? are they different between regions, between South Indians, North Indians?
Yes, yes, definitely. Definitely, we will be looking at it. Like we will. It's very obvious because you can find that it will also be influenced by the dietary pattern. So starting from the cooking oil, so it's going to be very evident that there's going to be a difference based on the regions, as well as the markers, the frequency distribution of the gene markers. So just like the one I mentioned. So that will also, the frequency distribution will also be different in different regions. So we have almost like 5,000 reports, the data. So when we analyze that, so we check that there is a lot of difference, especially North and South Indian. So when we check lactose intolerance, we could find gluten, and that could be probably because of more people started eating wheat products. So more of baked goods. Maybe I'm just like spilling out. And also vitamin D and B12 deficiency. So once you correlate with the biomarkers, but there won't be specific, vitamin D genes that are actually confirming this, but it's more of to do with the phenotype. We can just connect the dots, but definitely there are a lot of region-specific, differences that are observed, even based on the genetic patterns.
So that means it has to be region-specific and it has to be highly personalized approach. You cannot actually.
It was a lot of time, but we also should see how we can take this to a population-based, it's more of not a population, but we are just grouping them into genetic subgroups. And that again, we will be giving out, say, a similar kind of recommendation to people belonging to the similar subgroups.
Because in one of the reports of, I think, UK health report, I have come across their anticipation or expectation from gene testing is to make it applicable at individual as well as population level. So I don't know how far it is going to be realized, because it could be a dream, but if it is realized, it will be great.
Yeah.
It will be good.
It will take a long time for us to implement.
So I will just end up with the last question. Like there is a concept of tattva. In Ayurveda, there is a concept of following the treatment, including the dietary treatment, because Ayurveda gives a lot of importance to diet as well. Okay, so and they decide their approach is highly personalized approach, right from the beginning, and with all aspects, whether it is medical aspects or nutritional aspects. So they call it Prakriti wise, or Tattva wise. So do you think somewhere this gene testing is a scientific understanding of Ayurveda system? Can we say that?
Yes, ma'am. There are a lot of research studies that are happening in India. They call it as Genomics. So they have different, systems, one which you said, Tattva, and they group people, right? So based on certain Vata, Pitta, and all that. So that is also like the genetic subgroups. Based on the genotype, you're categorizing them. And it's based on the Ayurvedic principles. They're also, distinguishing on categorizing people. So where we could integrate both is we could actually find out whether there is some relationship between the Ayurvedic principles and the genetic markers. And then we can actually, so it will be in the future. So already in India and I think in one of the South Asian countries. So one physician was talking to us. I think he was from Thailand. So they are already into it. So it's not something new. But there are a lot of scope for it, Genomics.
The term Genomics, it's really very interesting. I think the previous gene mapping question has been clarified by the participant. His point is actually how you get a genetic makeup of a person, or what are the biomarkers or parameters to get genetic makeup to recommend personalized diet.
See, there is no standard thing, but only from my knowledge, like from my practice and experience. So usually what you do with the diet planning, you need information about the macros to give the macro distribution, micronutrients, so how much is your requirement. So you usually have a RDA and then you go with that. But here what you're doing is you're analyzing and taking into the consideration of the individual requirement by genetic tendency. So macros, micros, and if there is any allergy or tolerances, and anything else you want to actually decipher. So this is the usual protocol. So there is no fixed markers because it differs from a company to company, and it is also influenced by your affordability. The more the markers you test, the more you will pay. So the price will become more. So it's actually you who determine which genetic marker. So there are some companies which customize and then just check one marker for you and then say whether you're tolerant or intolerant, or whether you have the allergy or not. So there are some companies which will group all the disease risks together. So suppose if I want to know I have breast cancer because I have a strong history in my family, so I can just go and check for it, which is not actually related to nutrition, but it actually encompasses my health status. So even then it will be there, but there is no defined markers for nutrigenetic testing. So we are actually using with what is available. So just like you walk into any diagnostic lab, they have just coupled parameters into different packages. Let's say master health checkup, women's checkup, and all that. So it's just like they have certain templates like panels which they're using, and we are just making use of it.
Okay. So it is like, yeah, packages, right word you have put in. So you will not be like in a position to prescribe, like you get these markers tested so that you will be able to use that report. And
But
anyway, when it,
But that option is also available, ma'am. Some companies do give that option. So if we are customizing it, it will be again expensive. So it's rather best to go with the kind of the template that they have.
and your affordability.
Yes, that brings the cost down.
Yes, yes. Yeah, that is, and especially when it comes to diabetes, depending on the duration and the age of the individual or patient, you will be in a position to judge the proneness to various comorbidities and other health disorders, so that you will be probably explaining to him like, no, this is your proneness. So depending on the packages that are affordable to you, go for a test which helps us to understand your proneness to various other disease as well. Probably that may be one approach you may be following. Am I right?
Yes, that's very common. So we don't just stop with diabetes, but if they have other disease risk also, why not you check it those parameters as well instead of just sticking on to diabetes?
Yeah. And you must be even taking into consideration the family history of other health problems, right?
Yes, ma'am.
Thank you very much, Janvi. It is
a
wonderful session. I personally enjoyed today's session to a very great extent, interacting with you as well as Dr. Vimal.
Thank you, ma'am.
Wonderful for us to host both of you on our platform. And it's good that we got this idea of organizing this session as our contribution to the awareness of diabetes. It's very different from simply just telling people what is good to eat and what not is good to eat. So it's very nice. We appreciate ourselves also for accepting the request of our participants, and all our members are very, very happy with both the sessions.
Thank you, ma'am.
So much. Yeah. I now invite Neha to make certain announcements for today's sessions.
Thank you, Janani, for the wonderful session and the wonderful Q&A.
Yeah, thanks, Neha.
I'd just like to share some information with our participants for the certificates. So if the participants want to get the certificates, you need to compulsorily fill the feedback form and make sure that when you fill the feedback form, you write the correct email ID, because we use the email IDs that you enter while you are filling the feedback form to send your certificates. So it's mandatory to fill the feedback form to get the certificate. Feedback form will be activated at 7 p.m. today, and it will be active till 12 in the midnight tomorrow. So you please make sure that you fill your feedback form before 12 midnight tomorrow. It will take us 7 to 10 days to send certificates, and considering that it's Diwali next weekend, please do not send us an email inquiring for certificates before 20th November. If you do not get certificate for this session before by 20th November, please drop us an email to send asking about the certificate. Before you send us an email asking for the certificate, please do check your spam or promotion folders whether you have received the certificate from us or not. Thank you once again, everybody, for joining us on the session. One last thing, we are conducting a survey to study the effectiveness of webinars in enhancing knowledge, the link for which has been sent to all of you all in the mail today morning. So if in case you have not filled that survey, please do fill that survey form for us. Thank you so much everyone for joining us on this Saturday evening. Have a great weekend and stay home, stay safe. Good evening, everybody.